Okay, cool. So thank you, Yuri, for inviting me to give this talk. Hello, everyone. I'm Ken Hoehn. I'll just jump right in. So all of the work I'm going to talk about today focuses on B cells. So B cells are a crucial workhorse of the adaptive immune system because they ultimately produce antibodies. For me, they're especially interesting because they are really an evolutionary system. So like T cells, B cells also undergo VDJ recombination to produce the B cell receptor, but they have an additional diversifying process. So as part of the adaptive immune response, B cells undergo a process called affinity maturation, which is a process of rapid somatic hypermutation and antigen-driven selection. Basically, the B cell receptor mutates at a very rapid rate, and the mutations in the B cell receptor that increase binding affinity are selected. So this process of evolution by natural selection occurs within lymph nodes and within the body and functions to improve the binding affinity of the B cell population. It also produces what we call B cell clones, which are B cells that relate to each other by descent from a common naive B cell. So these two B cells would be considered part of the same clone because we can trace their ancestry back to this common naive B cell. So one of those useful tools we have for understanding B cell populations is the adaptive immune receptor repertoire sequencing. So in this, we just sequence the VDJ region of the B cell receptor. This can give us millions of reads in bulk sequencing or tens of thousands of cells for single cell sequencing. In single cell data, this can be paired with RNA-seq to give us gene expression information and basically gives us a way of taking a snapshot of B cell diversity within a sample. So the focus of this talk is B cell lineage trees. Now, B cell lineage trees are models of the ancestor-descendant relationships we see within the tips of the tree, which are our observed BCRs. So the way they work is we start at the root of the tree, which is our unmutated naive B cell ancestor. And the branches represent the number of somatic hypermutations that have occurred. And the branching pattern of the tree itself represents these of immediate ancestors. So these trees are just like what we would see with like phylogenetic trees of humans, with chimpanzees and gorillas, except they represent B cell evolution within a person. Now, one of the most useful things about B cell lineage trees is they provide a historical axis to understand different types of B cell diversity. So if our B cell trees contain samples from different biopsies, they can be used to study migrations between tissues. If they have different cell types, they can be used to understand cell type transitions. And if they're sampled at multiple time points, they can be used to study B cell evolution over time and reactivation. So for the first part of this talk, I'm going to be focusing on the first two, which is connectivity between tissues and cell type transitions. So just to make things clear, the goal, or perhaps the dream, I should say, of these methods is to be able to take BCR sequences annotated by tissue, cell type, or isotype, build our trees, label the tips by whatever piece of information we're interested in, and then use these tree structures to infer differentiation or migration networks. So this is a complex problem, and it's often the case that complex problems in statistics are first investigated using simple summary statistic-based approaches. So rather than jump into a fully fledged model, we decided to first start out with a maximum parsimony-based approach to get an idea of what questions could be answered. The approach we developed works by first building our B cell lineage tree and labeling the tips by whatever information we're interested in. In this case, we can just say some generic cell subtype 1, 2, and 3. So these are B cell subsets. We can then use a maximum parsimony algorithm to reconstruct the series of intermediate steps within the tree that minimizes the number of cell type transitions. So this would be an example of a switch from cell type 2 to cell type 1 within the lineage tree. And once we have that, we can compare the proportion of cell type switches that occur in a particular direction to the same tree, but with the tip labels shuffled among the tips. And this can give us an idea of the null background distribution of cell type transitions. An effective test allow us to build up a distribution, which is between cell proportions to our muted cell. So this positive bias in this distribution indicates that there's a greater proportion of cell type switches from 2 to 1 in our observed tree versus our permuted tree. And this test, which we call the SP test, can allow us to start using tree structures to identify ancestor-descendant relationships within the trees. One of the most interesting applications we found for this method was in studying B-cell migration during myasthenia gravis. So myasthenia gravis is an autoimmune disease caused by autoantibodies binding to the neuromuscular junction, causes progressive muscle weakness. It's often treated by removing the thymus, which is believed to be the maturation site for pathogenic B cells that release these autoantibodies. This is weird because B cells really aren't supposed to be in the thymus in the first place. However, though myasthenia gravis is treated by removing the thymus, remission can often take years or sometimes even a decade to occur. And one model for why this happens is that while B cells may mature in the thymus, some of them can escape into the blood, such that when we remove the thymus, we remove the source of these pathogenic B cells. However, some of them, which escaped from the thymus, can remain in the periphery for a long time and continue to cause symptoms. So we tested this hypothesis by obtaining BCR sequencing from 4 myasthenia gravis patients, both from thymus and blood sampling at the time of thymectomy. We found a substantial number of clonal lineages in both the thymus and the blood, and this is a representative lineage showing a lot of diversity in the thymus with some blood sequences at the tips. And if we perform our SP test on these trees, we can see in 2 patients a very clear enrichment of switches moving from the thymus to the blood. And this supports this model that these pathogenic B cell lineages really initially mature in the thymus, and then some of them escape out into the blood. And this suggests that B cell depletion therapy, in addition to thymectomy, so like rituximab, could be used to better treat myasthenia gravis, or perhaps early targeting of the thymus before these pathogenic B cells can really escape. So the next thing I'd like to talk about is how do we analyze time in the context of B cell lineage trees? How can we incorporate temporal information when we have the same lineage sampled serially through time? So one important question we can ask when we have this data is whether a B cell lineage is actively evolving or actively accumulating lineages over a sample interval. So this is an example of a measurably evolving lineage, where the earliest sampled time points are very close to the root of the tree, but the later sampled time points are much further away from the tree, or from the root of the tree. They're more muted. We can visualize this using a scatter plot where the x-axis is the sample time that we take the B cells at, and the y-axis is the divergence from the most recent common ancestor, which is the total amount of somatic hypermutation per site. And we can see in this lineage, there's a very clear positive relationship between sampling the lineage later times and there being more mutations. And we can calculate the significance of this relationship using a clustered permutation test, which we developed in this paper, to show that the correlation we observe between sample time and divergence is much greater than what we would expect from random chance. Now, since we can find lineages that are measurably evolving, we can also find lineages that are not measurably evolving. And this is an example of one of those. There's really no clear relationship between sample time and divergence. So this is basically randomly scattering the time points in the tree. And our permutation test reveals that this relationship is very non-significant. Now, this is an important thing to measure because one thing we expect successful immune responses to do is to stimulate affinity maturation. So if we give someone a vaccine, we hope that that is stimulating B cell lineages to enter into germinal centers, undergo affinity maturation, and produce evolving lineages. Of course, if this doesn't happen, we might end up with B cell lineages that are not evolving at all. And this brings us to one of the most challenging viruses to vaccinate against, which is seasonal influenza. So the most important public health tool we have for controlling seasonal influenza epidemics is vaccination. However, seasonal influenza vaccination generally has poor or varying effectiveness ranging from 20 to 60% in different years. And one model for why this is that pre-existing memory actually interferes with our ability to mount new responses to influenza vaccines. So that the idea is that when we give someone an influenza vaccine, we're actually stimulating memory B cell lineages, most of which immediately differentiate into plasmablasts, which release low affinity antibodies for past influenza infections. We're just re-stimulating past memory. And that this process inhibits affinity maturation against vaccine antigens. So basically, our pre-existing memory interferes with our ability to mount new, more effective antibody responses. So this is really a question of measurable evolution. If we give someone an influenza vaccine, can we detect ongoing evolution after that? So to test this, we surveyed measurable evolution in B cell data sets from 99 human subjects from 21 different studies, 3 of which were from flu vaccination, and most of the others were from other studies like HIV infection or healthy controls, West Nile virus. And this allows us to survey what conditions produce B cell evolution. So this is the result of that study. So each of these points is a different study and data set, which is colored by the condition. The y-axis shows the percentage of lineages in that study that are measurably evolving. And the x-axis shows the total sample interval, ranging from 4 weeks to 14 years. One thing that it really pops out is that HIV is really a standout in its ability to stimulate ongoing B cell evolution. It's a very strong signal, much more than anything else. The next highest, which is quite interesting, is children under the first 3 years of life, which I guess makes sense if we consider that kids are always getting sick and they're undergoing primary immune responses. So that's an interesting kind of unexpected result. But if we look at influenza vaccination, we find a very unimpressive amount of evolution. This suggests that lineages in the blood after flu vaccination just aren't really accumulating more mutations. Now, of course, this is in the blood, and the blood is not where affinity maturation is occurring. Luckily for us, we have really fantastic collaborators, such as Ali Elabedi's group at the University of Washington. And they're able to give patients influenza vaccines and then directly sample their lymph nodes using fine needle aspiration. And this allows us to really ask, are germinal center engaged lineages measurably evolving after influenza vaccination? So this data set samples the blood and lymph node at 5 time points after vaccination. It includes tens of thousands of single cells and monoclonal antibodies experimentally confirmed to be vaccine specific. And this allows us to really pick apart the influenza response in humans. And what we find is that if we look at lineages that contain both blood and germinal center samples, we find that lineages that only contain blood sequences are very rarely measurably evolving, but lineages with a high proportion of germinal center sequences are much more likely to be measurably evolving. So more germinal center cells, it equals more evolution. And this is one example of one of those evolving lineages. So we can see this lineage was sampled early in the blood plasmablast compartment, and then later at 60 days in the germinal center with a much higher level of mutation. This also contains a flu-binding monoclonal antibody, and these results really show that, perhaps contrary to what we may have thought, flu vaccination does stimulate pre-existing lineages to enter into germinal centers where they undergo additional evolution, presumably against influenza vaccine antigens. So to make all of these methods more widely available, we recently released an R package named Dowser, which implements all of the methods that I've talked about. If you're interested, check out dowser.readthedocs.io. It allows you to build trees, analyze discrete and continuous traits along B-cell lineages. Of course, it's named after the practice of using a Y-shaped stick to make questionable geographic inferences. I like to think that what we're doing is a little bit Better. And this is, of course, part of the incantation suite of BCR and TCR analysis programs, which allows you to start from raw data off of your sequencer and go all the way to analyzing your data, getting results, and making figures. So if you're interested in doing BCR and TCR analysis, check out incantation.org. We have a lot of documentation for our packages as well as tutorials. And with that, I'd like to thank you for listening. If you're interested in Dowser, again, this is the URL for it and the URL for incantation.org. I'd like to give a big thanks to our funders throughout the years, as well as my supervisor, Steve Kleinstein, and our collaborators. If you're interested in learning more about discrete trait analysis, this paper was recently published in PLOS Computational Biology here. And if you're interested in B-cell evolution over time, especially in the context of human influenza vaccination, this paper was recently published in eLife, and it's available here. And with that, I can take questions.
Well, thank you very much. Any questions to Ken? Please feel free to step to the microphone and ask a question. We have a minute or so for questions. Please.
Great talk. So I was wondering if you've considered the role of lineage-defining TFs in the extrafollicular plasmablast versus pre-GC, GC route. So we know that IRF4, IRF8, BCL6, and BLIMP1 drive the primary bifurcation. So if you reprogram these lineage-defining transcription factors in either, cell lines or in a murine model, do you think the evolutionary lineages you see would be affected, and how would it be affected by reprogramming these TFs?
So if you were to take different genes within B-cell lineages, either in some sort of model system, and then reprogram how they differentiate into different B-cell subsets?
Yes, one would bifurcate towards extrafollicular plasmablast, the other would bifurcate towards germinal center, towards somatic hypermutation, affinity maturation. So,
yeah, I think the most obvious difference is that if you just take a B cell and then differentiate it into like a plasmablast, I wouldn't expect there to be much mutation. Maybe if it undergoes an extrafollicular SHM process, then you could expect to find mutation there, but you would probably end up with a very flat tree. Whereas if you stimulated B cells to become germinal center cells, you would expect somatic hypermutation to happen and for basically B cell evolution to occur. So you would expect a branching and generally quite interesting tree to come about.